How to select the upstream HCP reference sample? Is it appropriate to take a single upstream HCP sample, or should a downstream be used as well?
So how do you select your HCP reference sample for generation of the antibody and for use in the immunoassay? So there's a couple different ways to do that. The reference sample should be chosen, should be representative of your upstream bioprocessing step. Generally, it would be considered to be a crude fraction, maybe sometime initially post-harvest, but not too far into the bioprocessing workflow. The second part of the question, which was whether or not a single upstream sample was suitable or something also downstream that might be more representative of the enriched contaminating host cell proteins, is appropriate. And there's not a single answer for that question. If the resources are available, it's probably good to do both. So an upstream reference material is going to capture more of the contaminants and is a good representation of what's happening in the bioprocessing and if there's any issue with the bioprocessing workflow. Having a downstream sample can be useful for detecting those more specific host cell proteins that may become problematic as the material becomes more pure during its bioprocessing workflow.
What are the animal species used for making HCP reagents?
The animal species used for making anti-host cell protein reagents most commonly is rabbit or goat. They've been used historically in almost equal amounts. Both are suitable for generating good broad coverage against all of the HCP proteins, where broad coverage is defined as covering all molecular weights from the very small proteins to the very large proteins. Typically for the animals, multiple different animals are used to increase the diversity of the immune response, because any single animal alone will not make antibodies against all of the proteins that are required. And so having a diversity of numbers of animals of either species is very important. And then in certain cases, again, if the resources are available, one could make reagents using both species and then have the option to pick the one that worked best or provided the best coverage against the host cell proteins. It's worth mentioning that another species is now being used more frequently, that is chicken. The rationale for using chicken is that it is evolutionarily more diverse or unrelated to the mammalian cells that are used to produce the biopharmaceutical, and it offers the ability to create a more robust immune response against more targets. It hasn't been used too long at this time, so it's difficult to say whether or not that's true.
What are the ways to improve or ensure good coverage of the produced anti-HCP reagent?
So generating anti-host cell protein reagents can be a challenging process. There are certain protein species within the sample that are poorly immunogenic or at low abundance and oftentimes do not give a good immune response when analyzing by Western blot. And so there are a couple of different ways to approach resolving that problem. One of them is to fractionate the protein into high and low molecular weight species and then immunize one of your animal cohorts with the low molecular weight fraction. What that can do is give you an enhanced immune response or enriched immune response against those low molecular weight proteins specifically. And that can then be combined back with the antiserum that's been generated against the total HCP reference sample. Other things that have been done is a technique called cascade immunization. Cascade immunization is one whereby an animal cohort is immunized with the host cell proteins. Frequently what will happen is there's several immunodominant species of proteins that will demonstrate very good reactivity in Western blots. What can be done is they can be created, purified, and then used to make an affinity matrix. That affinity matrix can be then used to deplete the host cell protein reference standard of those immunodominant protein species, and then the leftover material can be concentrated and re-immunized. That can be done a few times, and what happens over time when it works right is that you start to develop an immune response to the poorly immunogenic proteins.
What techniques, for example 2D Western blot or DIGE, are used to qualify HCP antibodies?
So, 2D Western blot or DIGE is a technique where the HCP proteins are separated based on their isoelectric point and their molecular weight. And that's important because there are so many protein species present in the sample. A simple 1D Western blot isn't sufficient to see whether or not you're detecting all the proteins which may be masked underneath other proteins. So the way to approach that problem is to separate them using this two-dimensional technique. It can be measured by Western blot, where you're demonstrating how many of the existing host cell proteins present in the sample can be detected by an antibody. And then the DIGE technique, very similar to Western blot, uses a fluorescent label and can also be used to determine how many of your antibody, how many of the HCP protein species are detected by the antibody. The coverage component is really an arbitrary measurement, and it's meant to be an indication of the quality of the coverage. General ranges can be 60 to 80%, and the number really needs to be a demonstration of coverage across the whole entire blot. So it can't just come from one single area. So as long as your coverage is across most of the protein species over all ranges of isoelectric points and molecular weights, and your coverage is above 60% or approaching 80%, the antibody is probably a good reagent.
How is an anti-sera pool made?
Frequently when producing an anti-host cell protein antibody, multiple different animals will be in a cohort set for generation of anti-sera. Not all of the animals are going to be responding in the way that's needed to produce the best possible reagent. Frequently, a selection of a subset of the sera collected from those animals will be used. So the way that that's done is analytical testing is done. That can be 1D Western blot, and then ultimately two-dimensional Western blot or DIGE experiments will be done to demonstrate that the reagents have good coverage. Those reagents that are sufficiently good to get you into the 60 to 80% coverage range across most of the different types of protein species can be pooled together to create a super pool. That then creates a single lot of anti-sera that can be used for downstream of all of the other things that are needed, including purification and assay development.
How is a mass spec being used as an orthogonal method for HCP analytical work?
Recently, an orthogonal method has been added in addition to 2D Western blot as a way to validate anti-host cell protein antibodies. And mass spec has some advantages in that it does not require immunogenicity of the protein species in order to detect it. So in that sense, it's unbiased at the different anti-host cell protein species. What it's being used for frequently now is to characterize the upstream HCP harvest sample in the bioprocessing stream to identify what are all the species that are in there. Subsequently, it's now also used to validate the anti-host cell protein reagent to complement the 2D Western blot experiment and say what specifically is in the sample, and also verify that the coverage is acceptable and good.
What is the best method for purification of the HCP antibody reagent? Purification by protein A or G, or affinity purification?
A frequent question that often comes up about anti-host cell protein antibodies is what to do with it once the anti-sera is generated and qualified. There's a couple of different decisions to be made, and there's a lot of arguments in the field about what's the best choice to make. One is protein A or protein G purification, which is used to isolate the IgG fraction of the anti-host cell protein antibody, and the other purification method is affinity purification against an HCP reference standard affinity matrix. There's positives and negatives to both, and there is neither one of the procedures is thought to be superior to the other. Protein A has the advantage of being unbiased in its selection of antibodies. The disadvantage of protein A is that it does not enrich for your anti-host protein antibodies, but also carries over all of the non-specific antibodies and can lower sensitivity of immunoassay during assay development. The advantages of affinity purification are that it can enrich for anti-host cell protein antibodies, so most of the things that are going to be reactive in your final antibody purified material are going to be reacting to anti-host cell proteins. It has the potential problem of being very biased towards the highly immunogenic proteins and may not purify equally across all different versions of antibodies for all the host cell proteins. So it's really kind of a choice of the end user on what they think might work best for them, because both have positives and negatives and both are acceptable methods for creating the reagent when submitting to the regulatory agencies.
Does the HCP antibody reagent support a single process or a platform process? If platform, how many products can it support?
So the anti-host cell protein antibodies can be used to support single processes. In the case where there's a new product being made and it requires a special process, in that instance, it may be able to use a generic or a custom HCP reagent. And in that case, the reagent can be made and tested. The point here about single process versus platform that's important to know is if the reagent is going to support a platform, it's very important that a large amount of material be made to support the platform, as it's unknown generally when it's first started how many different programs it's going to support. That means that if it starts off with 3 drug programs on that platform, it's pretty easy to determine how much of the anti-host cell protein antibody you'll need to support that over the long term. However, if that platform expands out to 5 or 6 or more drug programs, you could very quickly deplete that antibody material. So it's really important in the decision making about how much material you need to make for the long term to make sure that you don't run out of that critical material. Remanufacture and requalifying can be a time-consuming and burdensome process that no one wants to go through.